Well, good morning. I think we'll get started now. I'm Paul Danis. I'm with the consortium, and thanks for joining us today. We're excited today to have some different approach on this Proteoform Thursday. Usually we have researchers present the latest cutting edge work from their laboratories, but today we're having a chance to hear from some of our supporters in the community, like what are the latest technologies and applications that they're bringing out. A lot of this came to be like at ASMS, you see that a company will release like 7 different platforms on 8 applications. And it's really exciting, but I thought it'd be useful just to kind of have a focus on top-down applications and technologies and see really what's coming from there. So we're going to go ahead with this today. Let me give just a quick intro on a couple things. First off, thanks to our annual sponsors, Thermofisher, Bruker, Cyax, Lilly, as well as Agilent and Pfizer for supporting our activities and allowing us to have these kind of things. So we appreciate their support. And so if you see these folks, pat them on the back, say thanks for supporting Top Down. We really appreciate it. Also, just a quick note, if those of you looking to find us, we have a new URL, ctdp.org, if you want to find us on the web. And also, just as a note, it's still free to join the community if you'd like. You can hear about these activities, get links to past presentations and things like that. So go ahead and sign up there if you'd like, and we'll keep you informed as to what's going on in the world. Exciting next month, we have Oliver Hale from School of Biosciences at Birmingham. He's going to be talking to us about a little bit different approach where they're looking at intact proteoforms and native complexes directly out of tissue. So that should be really fun to see. So there'll be more details on that coming out shortly. And we'll have a wonderful time next month with Oliver. If you do have work you'd like to present, if you have some interesting work or you have a colleague that you think would like to share their work with the community, then you can just drop me a comment in the chat, or you can send an email to office@tdpro.org. Say, hey, I'd love to talk to you about my work or things like that. And we'll see if we can get you scheduled up for one of our Thursday events. So that said, we're lucky today to have Guillaume Tremintin from Bruker, Rachel Franklin from Agilent, and Amanda Lee from Thermofisher Scientific to present some of the latest technologies and applications from their world. So each one will speak about 15 minutes or so. We'll have time for Q&A after each of the folks speak, and then we'll go there. So I'll hand it over to you, Guillaume. You can go ahead and get us going here.
Okay, yeah, thank you for the introduction and the opportunity to present. So for the topic today, I chose something that came to us from Phasmatech, which is the team that's also developing the OmniTrap. And so I'll tell you a little bit about the Omniscape software, which is a new software product for de novo and sequence verification of top-down data. So I'm sure I preached to the choir here, but it's still pretty difficult to make top-down mass spectrometry broadly accessible.
We're not seeing your screen, Guillaume.
Oh.
I know you're having a wonderful conversation there, in your office, but the rest of us would love to be part of that.
Yeah, no, I would like to share indeed. Let me try to reshare again. Is that hopefully coming through now?
That's great. Thank you.
Okay, awesome. So yeah, as I was saying, it's still a bit of a challenge to make top-down data processing accessible to everyone. Several difficulties still around. The fact that many different ion activation techniques are used means also many different type of ions to take into account. Spectral complexity can be pretty high, so we have to deal with like very congested spectra, overlapping isotopic clusters, and so on. And probably the hardest part of all is the fact that we have access now to this rich array of proteoforms, and we can see them exactly how they're distributed on the protein backbone. But that, of course, makes database searches much harder. With a top-down, with bottom-up, we can easily catch a few unmodified peptides to at least get to some level of identity. This is more difficult in top-down. So the design goal behind Omniscape was to have something quite accessible that would enable de novo sequencing. Main purpose here being to identify a known protein, so you have something to start with. Based on that initial step, have sequence confirmation tool in order to progress from this initial identification towards actual proteoform level information, and also to offer possibilities to combine various results together in order to build as much of a picture as we can of the protein of interest. So the workflow initially looks like this. The software was designed to offer a lot of versatility in terms of the data input. So you can bring in actual MALDI data with something like the NeoFlex, for example, and MALDI in source decay, regular QTOF data, data from the timsTOF series, like the regular and the ultra, as well as MRMS, and eventually Omnitra-based instruments. So for all of these platforms, we can use data analysis software to collect the peaks we're interested in and create collections of MSMS spectra that we want to process. At this juncture, we import those data into Omniscape, and this is also the opportunity to bring in data from third-party instruments. So the software is in principle open to just about any kind of high-resolution data, and as long as you can get it into a text, you can get your spectra in a text-based format, you're able to bring them at this point as well. So you're not limited to the Bruker world. And finally, we do the processing into the Omniscape software. So I'll quickly take you through what that looks like. The initial step, regardless of the workflow, is that isotopic cluster identification. As I mentioned earlier, we often have very congested spectra, overlapping isotope, and to have any chance at getting a good output, we need to clearly identify which ions are actually in our MS2 spectrum. So this is something that we can facilitate on all of our high-resolution QTOF data. We have what we call this true isotopic pattern, which allows us to get a very precise representation of isotopic intensity, which in turn make life easy when it comes to de-isotoping. And what's new here is the OmniWave peak-picking algorithm, which applies a few novel tricks to perform the de-isotoping and the decharging of the data. So that allows us to build mass lists with very high confidence. From a user perspective, what this looks like, if you start from an unknown MS2 spectrum, you can choose the de novo workflow. Let me grab the pointer over here. You can choose the de novo workflow, load in a data set, or here I just have the illustration of one, but you could also get multiple spectra here. Choose basic parameters such as mass tolerance, ion types, and so on, and start the workflow. The software will go through the steps I mentioned earlier. Look for related charge states, discharge the data, look for isotopic cluster, create such a collection, and then look at distances in order to build to build sequence tag. So the sequence tag can be created using a various number of roles. You can set a number of criteria in terms of charge state relationship, in terms of ion complementarity, and so on. Once you have those kind of collections, the software can automatically export a string like this, which is ready to search into a public or private BLAST server. Hopefully the outcome of the BLAST search is that those sequence tags are sufficient to identify the protein, ideally uniquely identify it. So here was just a model with carbonic anhydrase. So we do have the highest score for the bovine type, which is what we had used. And at this point, that allows us to get at least a starting sequence for the protein. And if we're lucky to work with an annotated database, we can also retrieve information about expected PTMs, processing, and so on. So once we got that, we can easily go into the sequence confirmation workflow now. So we can reload our MSMS data. Similar set of parameters initially in terms of tolerance and ion type, but we now can input a sequence and associated modifications so that we can calculate a list of expected proteoform. And this is extremely fast. So we can really go fairly crazy on how many proteoforms that we want to experiment with here. Once we start the workflow, so just a quick highlight here to show you that you can configure the sequence, so you have the ability of adding any fixed or variable modification as needed for the problem at hand. So you have a lot of flexibility there. And once the search is through, again, you generate your pick list and compare those pick lists against the possible proteoform to come up with a notation. So here we're looking at the highest scoring proteoform. The score is a combination of sequence coverage and intensity coverage, how much of the signal in the spectrum we're able to explain with a particular identification. And you can visualize a little fragment in a fairly traditional sequence map view, where you get your sequence, your N-term fragments on top, and your C-term fragments on the bottom. So you can quickly see the type of ions that have been annotated, as well as the coverage. There's a number of parameters you can look at for the QC of the data. You can see those ion intensity maps. So here we see that we get most of our C ions from the N term and most of our Z ion from the C term, which we would expect, but that gives you a sense of the data quality. You can also look at the charge-state relationship. So here, I'm sorry, I clipped the x-axis, but the idea is that we look on the x-axis at the length of the fragment and on the y-axis at the charge state. And of course, the expectation is as the fragments get bigger, we get more charge state distribution as well as just a higher average charge state in general. So we get that X pattern. So again, that helps us confirm the quality of the annotations that we have. We can look at the mass error on those fragments as well. That allows us to get a global view of the data quality, the data annotation quality. But we can also click individually on any of those fragments to view the actual raw data overlaid with the theoretical isotopic distribution for that particular fragment. So by clicking through the data, we can very easily get a sense of whether or not we trust the assignments based by the software. And from there, we can select, deselect fragment in order to really only keep a collection of things that we truly believe represents the sample correctly. As I mentioned earlier, we can then take those multiple results. So we can generate a collection of sequence map like this using different charge state, using different type of fragmentation, whether we go with some kind of EXD mode, CID, in-source decay, and so on. We can also combine MS2, MS3 levels. So this is particularly useful, for example, for MODI in-source decay, where we generate a global MS2 spectrum. And then we also generate an MS3 spectrum, which is focused on the N and C termini in order to refine the coverage on the termini. And we can also combine collections from diverse instruments. And using all of that, we can boost the sequence coverage and boost our confidence in the PTM assignment for each proteoform that we assign. So that's what this looks like. So this was data acquired on a prototype team stuff, where we got 86% on the NISMAB light chain using ACD, 85% on the NISMAB light chain using EID. But combining those two, we can actually increase the sequence coverage to 94%. Reason being, with that EID fragmentation mode, we can fill in a little bit the middle part of the sequence here, which is not as rich when looking at ECD. So that gives us the ability to combine experiments in a convenient way and really get a high confidence sequence. So with that, I hope that, brief presentation was useful to show you a little bit how we have now a user-friendly tool to analyze your complex top-down data. I think the de novo part in particular is pretty unique and really extremely useful if you're working with spectrum that do not work with standard tools. We've seen quite a few examples of that already. Very flexible sequence confirmation tool, including the ability to really visually check the result overlapped with the raw data, so you can build confidence. And generally speaking, the entire software is extremely fast. So I didn't play a video here, but this entire little workflow I was showing can be performed in 3-4 minutes. It really takes very little time. So yeah, with that, we aim to provide an easy workflow to go from an unknown spectrum all the way to a proteoform analysis, putting you in control of looking at of the data, making sure you trust it, and maintain a very open ecosystem like we've been doing with all the TeamStuff product, and make sure that you can bring data from anywhere that makes sense. And with that, if that seems interesting, we do have also a webinar next month with Marie-Angela, who's part of the FastMatech team, and came up with a lot of the tools that I showed today, as well as Arndt, who's one of our multi-TDS experts. And yeah, they'll be able to tell you a bit more about what we can do with all this. And with that, I'd like to finish. And again, just acknowledge Marie-Angela, Detlef, and in general, the whole Omniscape team that have been doing a fantastic job putting this together. Yeah, happy to take any questions.
Great.
Thanks for the attention.
Actually, thanks, Guillaume. Fascinating. It's great to see. We do have a few questions in the chat, so let me go through some of those. Nina's asking, how often do you check fragments visually to confirm? Is there any criteria?
Well, I guess it depends a little bit on the. It depends a little bit on the purpose of the analysis. Like for me, I mostly come from the antibody side of things. So if I'm making a call on a particular degradation product or something like that on an antibody, I would definitely want to just visually look at it, just to be sure of what I am claiming. I can appreciate in a more global top-down approach that might be a challenge to do so.
Okay, great, thanks. Steve's asking, is the MS BLAST algorithm directly derived from the NIH's BLAST modified version or something completely different?
No, the BLAST is completely standard. So we just use the regular software.
Okay, thanks. And Nikita's asking, which type of fragments are available? Can they work with different DWV internals?
Yes, so I did not mention that in the presentation, but we support the regular internal fragments. So I think I can go back quickly over here. So you get those traditional paper fragmentation, but then you get those like internal those various internal ions available as well. And this has been already useful. We did get a few spectra already where we using normal ions had very poor sequence coverage, and for some reason found out that we were producing an incredible amount of secondary ions. So that's can be a very useful feature.
Sounds great. I think that would take care of the next question as well about internal fragments. Any other questions for Guillaume? All right, great. Thanks, Guillaume. So next we'll move over to Rachel Franklin from Agilent. It's all yours, Rachel.
Thanks. I'll share my screen. Okay. Hopefully you can see that.
Looks great.
I'm gonna hide my top panel really quick. Okay, thanks everyone. My name is Rachel Franklin. I'm an application development scientist at Agilent Technologies. And today, of course, I'm going to tell you about our latest tools for top-down characterization, but I'm also very excited to teach you about an important protein engineering technique, which is increasingly relevant for the top-down field. Okay. So there seems to be like a constant flow of hardware and new software techniques for top-down characterization. And I think an example of that is even just in the past presentation with the new Omniscape software for top-down de novo sequencing. So with all of these kind of new techniques, how do we go about validating top-down results, specifically for proteins with post-translational modifications? And the problem really is that we don't have a way of creating standards of post-translationally modified proteins. And historically, it's been challenging, if not impossible, to do that in a site-specific way. And so I'm excited to teach you today about something called genetic code expansion, which is an emergent protein engineering technique, which essentially makes designer proteins with made-to-order chemistry in the exact spot that you want it. There is an amazing center at Oregon State University that is funded by the NIH to help develop tools to get this into the labs of other folks. And the key parts of this is the orthogonal translation system, which helps put a non-standard amino acid, so one of the non-standard 20 amino acids, into a protein at a very site-specific spot. So that can be something like phosphoserine, for instance, which essentially makes a site-specifically phosphorylated protein. So why am I telling you this today? And that's because actually combining genetic code expansion with top-down mass spec has immense potential to not only advance what we can learn about biology, but also advance the field of top-down mass spectrometry itself. So I'm going to talk a little bit more about that today. And first, just all of the results that I'll be showing today were acquired with the Agilent QTOF, specifically the 6545XT. And that was equipped with the brand new Agilent EXD cell, which is shown here in this zoomed in picture. And the EXD cell is located just directly to the collision cell. And because of that geometry, we can do both ECD, CID, or both within the same experiment. And then also the 6545XT was designed for protein analysis. So it has an ultra-high vacuum and the ability to isolate precursors out to 4,000 m/z, which is really important for top-down experiments. And finally, our analysis package for top-down fragmentation analysis is called EXD Viewer. And it's a great, very user-friendly software that helps you really get a good, deep understanding of the quality of your top-down results. And it is available for free, so you could actually download it today and try it out if you'd like. You can look at this link or probably just search EXD Viewer Agilent. Okay, so I mentioned that we can do CID, ECD, or both in the same experiment. So I want to spend some time first just talking about these different fragmentation techniques and the type of nomenclature you're going to see in the rest of the presentation. So collision-induced association, or CID, is by far the most common fragmentation technique that you'll encounter in tandem mass spectrometry. It mainly works by heating up proteins or molecules with energetic gas collisions, producing B and Y ions, which you see here. But it also often cleaves off-label chemical groups like post-translational modifications. And it has limited utility for top-down analysis. So in contrast, there's electron captured association, or ECD, which is complementary to CID. But this is a more gentle fragmentation process, which often preserves post-translational modifications like phosphorylation. So that produces fragment ions that are called CZ ions, and then sometimes some other types such as A and W. The W ions specifically are important because that helps us differentiate between isoleucine and leucine. And overall, ECD is much more effective for top-down analysis. And in EXD Viewer, this is what the annotation looks like, where the different N and C terminal ions are represented as different colored circles, where ECD type ions are in blue, CID is in green, and so on. Okay, so today I'm going to talk about two different applications where we've used top-down analysis to analyze genetically phosphorylated proteoforms. And so the first example I'm going to highlight today is looking at sequential signaling pathways. And the second example is focusing on protein complexes. And again, all of these are done with the tools that I mentioned previously. Okay, so first we'll start with sequential signaling. Okay, so sequential phosphorylation is a fundamental mechanism for cellular signaling. And it's been really difficult to try and piece together what the order of operations where one phosphorylation happens before another, largely due to the fact that we just don't have those standards with a very specific set of phosphorylations to test. So in this analysis, we turned to genetic code expansion to site-specifically engineer a phosphoserine amino acid into this protein. And this is the MS1 spectrum where you can see the protein molecular weight here, and it has an encoded phosphorylation. You can see this is very homogeneous signal. And with this very well-defined proteoform, we then mixed it with a phosphorylation-dependent kinase called GSK3 beta. And GSK3 beta is known to add several phosphorylations to this protein. And we see that here is a shift of mass in 560 Daltons. And so now it's really up to us to use top-down mass spectrometry to isolate this hyperphosphorylated protein and figure out where the sites of phosphorylation are. So the workflow that I'm highlighting is done in ExtViewer, and the first step in a phosphorylation site mapping experiment is just to define the target sequence. So here on the right, I have our target sequence, where this particular sequence has a total of 21 possible phosphorylation sites. So every serine and every threonine is assigned as either a fixed or variable modification, so phosphorylation in this example. But custom modifications can also be added, and that can be useful for other applications such as synthetic peptides. So once you set up your sequence and you search that against your data file, this is the results view that you can expect from ExtViewer. And there's a couple of key features that I wanted to highlight. So first is just on the left here is the mass to charge spectrum, where you see all of your fragment evidence. And we have the match of M to Z and also the intensity kind of illustrated here with this line that shows you the isotopic match. And every one of those signals is then represented in the sequence map, where again, the different colors are corresponding to the different fragmentation types. And this is extremely interactive. And so clicking on any one of these fragment ions brings you to where that sequence or that signal is in the mass spectrum. So in this example, all of the identified phosphorylation sites are in red text here. And at the bottom in the targets tab is a list of all the top scoring sequences. And so you can explore and kind of compare the fragment evidence for all of these sequences by just toggling through this list, or also opening up a side-by-side view and comparing it simultaneously. And there's a couple other useful graphs in ExtViewer. Of particular mention is the fragment intensity graph. And this is really important for helping you evaluate your data quality. So this tells you how much do you understand about your spectrum. So how much of the signal was corresponding to the precursor, how much of it was corresponding to those fragment ions, and how much of it is ion signal that doesn't match your target sequence. And there's a number of other helpful graphs that I'm not going to emphasize today. Okay. And while I'd love to spend a lot more time telling you about the work that we've been doing with sequential signaling, I want to make sure we have time to go over the second application that I'm highlighting today, which is looking more at protein complex formation. Okay, so site-specific phosphorylation is well known to modulate protein-protein interactions just kind of in a broad sense. And our collaborators at the Genetic Code Expansion Center have done this really elegant study where they've focused on this protein called BCL-XL, which is crucial for regulating cellular apoptosis. And it's known that phosphorylation of a very critical residue changes its binding interactions with pro-apoptotic proteins. And so we set out to create this BCL-XL proteoform and kind of test the downstream binding. So after we used top-down mass spectrometry to kind of in a quality sense to confirm we'd made the proteoform we thought and so on. But after doing that, we got interested to actually see what ECD could tell us about the intact complex. And so we took, in this example is BCL-XL with a peptide binder called BAD. And we just mix them together 1-to-1. And this is the intact protein spectrum, which you can see in monomer BCXL, but we also effectively retain some of that complex. And we isolated that and sent it forward to the EXD cell for further fragmentation. And the resulting fragmentation of this complex is actually a mixture of fragments from 2 different sequences. So we have the BAD peptide, but we also have BCL-XL. And that can cause some complications for top-down analysis. But EXDViewer does a really good job at kind of separating these signals in its multi-target matching feature, which helps analyze chimeric spectra. So after fragmenting the complex, these are the sequence maps that we obtained. And when we first saw this, we were actually really excited because we immediately noticed what seems as these large gaps kind of in the middle of the peptide and the protein. But when you map those fragments onto the crystal structure for this complex, you see that these gaps actually correspond really well to the predicted interaction surfaces for the complex. So this is evidence that these methods could even be used to gain higher order structural information, which is really exciting. Okay, so in summary, there's just a couple key points that I hope you take away from this today. One of them being that EXDViewer is a great software for top-down analysis of not only unmodified but modified proteins. It has an effective workflow for complex down fragmentation, and it's available for free for you to try out today. And I really encourage you to download it. And there's some example data files you can test out. And importantly, what I really want to emphasize for this crowd especially is that combining top-down mass spec with genetic code expansion gives us new opportunities to make proteoform-specific measurements. And I believe that within the next couple of years, this is going to be much more common in the field and will be really relevant. So with that, I wanted to leave you with just a couple exciting future directions for this combination of technologies. And I'm actually not going to read them out loud right now because I think I might be running short on time, and I want to make sure there's questions, but these are just for you if you're interested. And with that, thank you to everybody who made this work possible, and thanks for the consortium for setting up this event. And I will take some questions now.
Great, thank you, Rachel. Exciting stuff. So we do have a couple questions in the chat. Also, if you want to ask a question live, just raise your hand or just unmute yourself, and we'll be happy to just interact that way as well. So the question from Sandrine: how is the repeatability of the abundance of the C and Z ions? Is it useful for label-free quantitation?
That's a really great question. I can't say we have tried label-free quantitation with this. So it's definitely an area of future exploration that I would love to talk to you about if you're interested. This is kind of new. So I guess I kind of also skipped over this, that the Agilent EXD cell is new as of release at ASMS this year. So I'm always open to exploring new application areas with that.
Excellent. Always good to have new toys. Richard has a question that when you do the genetic code expansion to install 5 phosphomods on the same protein, what is the efficiency of installation, and does the fully modified protein need to be enriched?
That's a great question. And the efficiency is, it's very efficient. They get very little non-incorporation or premature truncation. So I'm working with some pretty pure abundant standards. And no, it doesn't typically need to be enriched. Yeah, so that's kind of the beauty of genetic code expansion, is having these really great authentic standards to work with.
That's great. I just follow up on that. Just curious, when you get one of these standards, say it's whatever it was, 20 or 40,000, and you have one site of phosphorylation, do you get an indication of like, is that like 90% pure on that one site, or is it like a 98, or is it 60? What's your sense of like the purity of these things if you get like a single phosphorylation?
Right. So just due to the mechanism, you would not expect to find it anywhere else in the protein. So I think the heterogeneity is not necessarily where the site would be, but if maybe the site's there or not. And there's also issues sometimes with premature truncation, but those have largely been overcome. So really, it's 100% what you think it is, with not very much variation at all.
Wow. Well, that's impressive. Cool stuff.
Yeah,
And
and they can do multiple, so not just one site, but multiple. And this is just an example of phosphorylation, but they're also working on all sorts of different amino acids, ranging from things that are much more exotic, like tetrazine for click chemistry, and it's all over the board. It's pretty exciting.
That's great. Thanks, Rachel. So Nina has a question. Terrific presentation. Does the EXT viewer account for scrambling and fragment ions?
Scrambling. I'm not sure if you could clarify. I'm not sure if maybe you're talking about HDX scrambling, or yeah, I'm not sure what you're asking for that. Sorry.
And Nina, did you want to just ask live, clarify what you're thinking there? Okay, well, we'll let that one be. I just a question on that. You're doing the complex work. Were those complexes also derived from the GCE technology?
Yes, they were. So the idea there was testing that with a specific site of phosphorylation that was known to kind of change the binding interactions of that peptide binding partner that I featured. And they actually went forward in that and did more different characterizations with different biophysical techniques, such as isothermal titration, calorimetry. So this was kind of a combination of techniques for this project.
Wow, very nice. Any more questions, Rachel? All right, great. Thanks, Rachel. Now we're going to hand it over to Amanda Lee from Thermofisher. So go ahead, Amanda, it's all yours.
Hi, thanks. That was a really great presentation, by the way, Rachel. Really cool incorporating that. Yeah, that was really awesome. Let me share. Oh, it actually did the right thing. Let's see if this works. All right. Okay. Okay, hopefully that's sharing properly. Hide that. Okay. All right, I'm sure you'll let me know if anything is going wrong. Hi, I'm Amanda Lee, product marketing manager at Thermofisher Scientific, managing for the Tribrid, Orbitrap Tribrid line. I'm going to introduce the newest in our line of Tribrids, which have been traditionally used a lot in the top-down field, to be honest. When I was in grad schools, when the Lumos first came out, just at the end, and got really cool data there. But so I'm going to just introduce the latest in that line, and then some of the applications that some of you may not have seen. They're a little newer, some of these applications. Some of the papers are newer, and some of the capabilities of this new instrument, the Orbitrap Ascend. And then some of it's also from an Eclipse, which is the previous Tribrid in the line. So mixed bag here. I'm hoping I can cover enough of this to get everybody interested. You can always ask me questions, message me. So just to introduce you to the newest product in the Orbitrap Tribrid line is the Orbitrap Ascend Editions mass spectrometers. We released these 3 application-specific editions of the Orbitrap Ascend. Multiomics, structural biology, and biopharma. Each of these application spaces has room for top-down proteomics or protein analysis, I'm going to talk about those specifically in this presentation, but just so you're aware of what we have. So what's new with this release? The biggest thing is number one, the native MS option. We've added new hardware into the Orbitrap Ascend that extends the isolation of precursors in the quadrupole. I'm going to talk a lot more detail about that in further slides, and just show how that's useful potentially in top-down and maybe in your experiments, or if anything you're interested in that you feel like you've been limited in. But that native MS option also includes previous available abilities on the Ascend, including extending the mass range up to 16,000 m/z. And the ion trap can still do its isolation as well. Both the ion trap and the quadrupole now isolate up to 8,000 m/z without compromising the low m/z isolation, because you can toggle between, and they use different sources for that. For the voltage. Anyway, other new things is TMT Pro 32-plex is new. We have new resolutions for that to support that. Those resolutions could also be used in top-down, I suppose, if you want. We also have specific method template locations per the different editions. So that means basically if you're in a multi-omics or structural biology or biopharma, your method templates are more organized per your edition. And the Ascend editions come with dry pumps for more environmentally friendly reasons. Skip through that. Okay, so then the native MS option. So what's new hardware-wise here? So in the previous edition, or the previous versions of the Ascend and the Eclipse, we actually call this high mass range, HMRN, and that isolation used the ion trap for the bigger m/z. So anything above 2,000 m/z. That was minimum you could go to 100 Thompsons. And so you can see in this little example that would not isolate proteoforms, right? In our new, sorry, no, there you go. In the new hardware, we're now able to isolate down to 5 Thompson windows in the quadrupole, all the way up to 8,000 m/z. This is allowing for more selectivity. It's also improves the sensitivity as to where the quadrupole is in the architecture of the system. And I'll show that coming up if you're unfamiliar. And so just like an example here that we wanted to see, NIST MAB is a great example of a big m/z in native that has proteoforms. So with this new quadrupole, we're able to isolate each of the glycoforms of the NIST MAB, the major ones at least, and select them for further fragmentation or whatever you wanted to do with them in any kind of manipulation. But I wanted to emphasize here the signal to noise increase. We can see it's quite large when we get the isolation, which makes sense. The isolation is efficient. In the quadrupole, it's our best quadrupole here at Thermofisher. That's in this in the Ascend. So we took one G0F, G0F glycoform for NIST and did multiple fragmentation techniques, which are options on the tribrid line. Combine those to give a coverage. You can see that there's really specific locations of the fragmentation that's indicative in native mode. This is a native infusion. Makes sense that the fragmentation is limited to the regions of exposure. Anyway, and we get a pretty good coverage in native form of intact antibody. I think can argue. And another application that I think is very interesting and something that we do see in top-down, and I'll talk about more later, is this DIA-PTCR. There's a few other names, and I'll talk about that later. But basically what we're saying here is if you have a very complex intact infusion or even LC, your proteins don't always separate. You have a lot of different proteoforms. They elute at the same time. This happens a lot in top-down. What we can do is take a narrow isolation window and perform proton transfer charge reduction, PTCR. It's a highly fluorinated compound that's internal to the mass spec that we can allow to react with our precursors. in that isolation window and spread out the signal, basically. It's not a fragmentation technique, so it's not like DIA you may have heard of already. It's stripping protons from your precursors so that you spread out the signal. And because you've isolated, now you have less complex starting material, basically. And so you can deconvolute that and get average masses of things. Or you could take that window and fragment those more separated now because you've expanded out the M over Z. So more separated precursors for further fragmentation, and that's what I'm going to talk about later as well. So just so you're aware, this is the architecture of the system. Their Ascend actually has a lot of differences from previous versions of the tribrid. It's the biggest architecture change we've had because we've introduced a front ion routing multiple that allows for storage of ions before the C-trap orbitrap region. So if you're doing more complex experiments like top-down does, where if you're going to do ETD in the back, or PTCR or UVPD or CID, there's a lot of fragmentation techniques. But if you're doing those in the back and then you have to send them to the orbitrap for MS2 or maybe even MS3 analysis, you can still be collecting your next precursor. As you know, sensitivity can be an issue. And in these systems, sensitivity is tied to how much time you spend collecting precursor, potentially, if you have a limited sample amount. So that can really help you boost that sensitivity by having your collection in tandem. So we have really good parallelization of those. Anyway, but benefit for top down in that sense. But another big benefit, sorry, is that we have the native MS option that extends the quadruple isolation, expands our detection. Those are really helpful, especially if you're doing native, which is something I'll emphasize in coming slides. So I'm going to start with the omics level information, just to full transparency. The information from these is actually from an Eclipse, so it could be done on an Ascend, of course. But I just want to talk about something that's fun and cool, that I thought, at least in a proteomic sense. So we have this really interesting work that was done with fractionation. So that can really help in the complexity of top-down proteomics. We tend to have a lot of different proteoforms and they can be really hard to separate out. So I'm going to talk about two different ways that we've tried to help with this fractionation. The first one being FAMES. So FAMES is ion mobility that we have on the front of our system. You kind of see it in the little picture here. It's almost like a filter in a way, and that really helps simplify your precursors coming out. So in this example, they had serum. They did some depletion, they did some separation. They're trying to get it as simplified as possible, but it's still really complex. So adding FAMES helped because, as we can see down here, using a couple different representative CV values, you can see how different at the exact same retention time, how different that precursor population is. That fractionation can be really vital in top-down because of how these would have co-eluted, and it may have been difficult to isolate any one specifically and get really good fragmentation because of the complexity. And now we've kind of limited that. So we got an increase of 24% actually of the IDs from serum proteoforms, specifically different proteoforms, using this technique. So that's really fun. Reference at the bottom. And then another technique of, I'm calling it fractionation. I believe it's a type of fractionation in a way, is PTCR, which I talked about before with that DIA PTCR. In this reference, the authors are calling it targeted T PTCR, where you can isolate, again, if you have a very complex MS1, a lot of precursors, a lot of proteoforms, you can isolate those and perform PTCR, proton transfer charge reduction, and expand out the charge range of the isolated region. In this case, they do really narrow isolation windows. The quadrupole is really good at that. So it's very possible to go very narrow, and now all the way up to 8,000, and get that expanded region, and then do MS to the third, which is actually fragmenting your precursor, so that you can get identifications. So combining those techniques, we have a huge increase, almost double pretty much, for larger proteins. And I remember starting in top down, there was this huge barrier almost of saying our limit is 30,000 Dalton size proteins. But using these sort of techniques and different fragmentations that are better for larger, we can really expand to bigger proteins and push the limits, which is really fun. So another technique, I'm moving into that structural biology. This is actually on the Ascend editions, the structural biology edition, with collaborators from Professor Dame Carol Robinson's lab. Doing native top-down, really interesting stuff. I think I love native top-down. In this example, they have a protein, they are fusing it, and the M/Z was above our previous limit of 2,000 M/Z for the isolation. So with the new quadrupole option, they were able to isolate their most abundant charge state, fragment it. They used HCD in this case. You could use whatever fragmentation technique you want and get really nice sequence ladder information, confirming your protein. If you're in an unknown situation, if you're infusing a known, you can start mapping where those fragments are, and maybe get some information on the structure a bit, any binding, anything like that, whatever's of interest. Just an example here. But really cool stuff. Multiple fragmentation techniques are possible for your native work. Okay. And then in the biopharma realm, this work was done on the biopharma edition, actually, of the Ascend as well. So in this case, they're doing middle down analysis, which is the IDES digest of an antibody, and then reducing it. So you have the light chain, you have the FC, and you have the FD region. In this case, they're trying to do antibody drug conjugates, ADCs. So we're trying to figure out how many and where the drugs are conjugating onto the antibody of interest. In this case, we can see separation. This, I believe, was on a MABPAC reverse phase column, very normal sort of setup. No crazy additives or anything like that. Separating out the regions or the different additions in the FD. Just highlighting here the one with 2 drug conjugates. They were able to map that by combining fragmentation techniques, so HCD, ETD, ETHCD, and UVPD. Any questions on any of those, please message me. I'll be happy to talk your ear off about all that. But anyway, so combining that and doing PTCR, which is a very interesting combination at the fragment level, where you can take very complex fragmentation. And as we know in top-down, because there's so many paths of fragmentation, you can get really complex MS2 or your precursor scans or product scans, sorry. Those are really complex, especially by the precursor region of the spectrum. So you can take those and isolate them to do another MS to the N stage where you do PTCR. Just like I was showing you with the DIA PTCR or the TPTCR, you can do that on the fragment level and then expand out the fragment ion pattern so it's less complex. You can do better deconvolution and get more IDs. That's in the reference as well. So we can always, I wanted to show so much more data, but time limited and I gotta hurry. So anyway, these techniques can really map out where your drugs are. That's similar to any sort of PTM. You can use more gentle fragmentation techniques if they're labile, just ETD. And that is a great technique for those labile PTMs like Rachel was talking about. So very interesting work. And in my opinion, I love top-down. I'll talk your ear off about it. Anyway, and so more with the PTMs here, and just so you can see different fragmentation techniques. This is not including the PTCR, but just to show you how you can get this different mapping, different PTMs. You can see where they live on the protein with your fragmentation pattern. That's just with ETHCD. Finding phosphorylation and how a labile modification can be retained well in this system. The Ascend has a gentle front end for that, and ETD is a gentler fragmentation technique, helping retain. And also how when you combine fragmentation techniques, like in the previous slide, why? So just to show you a representative chart of how orthogonal the fragmentation techniques can be, of how many more fragments you can gain by combining them all. And we saw that with the other vendors' talks as well. Combining techniques helps a lot. That's not a new concept, but just so you see. how interesting it can be. And I threw this in for fun, so sorry, indulge. But I know we're saying top down, this is generally proteomics, but there's also fragmentation available for oligos. And you can do top down, like fragmentation of intact biomolecule for oligonucleotides as well, using this low Q CID technique on our systems. That's really vital actually to lower the Q value for oligos, especially large ones, to avoid more difficult internal fragmentation. And it's very easy to change this in the method editor also if you ever have questions about it. But really fun technique as well, top down for something other than proteins. Just had to throw it in because I thought it was really nifty. Yeah, I want to take questions, so I'm hurrying. So thank you. Please message me. There's a recording, so my email's at the front of this. Let me know if you have any questions.
Great, thanks. Any questions for Amanda? I had one I was wondering about as I'm looking at, it's more not just for you, but for a kind of a general question, is like a lot of these platforms, they offer like so many methods within one platform. Now, if you walk up there, say with a $50,000 Dalton kind of normalish protein, is there like some guidance around the best practices as far as what you do first and second and third to unravel that? Or do you just kind of throw them all at it and see what you get?
So, well, at least for our platforms, we have template methods. So you can always use one of those as a starting point. It has all the settings that you would need generally for based off size and the fragmentation technique of interest. So you can start with those and then adjust in source CID or maybe the AGC or the injection time. You can adjust those as you see necessary. The templates are a good starting point though. To know which fragmentation technique to use, that's a little bit more up to you. Maybe you don't have all the options because the standard ones on our tribrid systems is CID and HCD. If you have ETD, that's a great one too. I always start with HCD to just personally. It's a really good technique and it's a nice one to start with because you normally see something because it's normalized as well. So it's a good one to start with. And then a template for ETD to know the reaction time approximately. We have the templates for those. So.
Okay, thanks. So not start from scratch.
Yeah, never start from scratch. Yeah. And there's lots of publications too. We can share.
Yeah, because it's one of the challenge for we hear a lot of people coming into the field, and it's one of the big challenges is the training and the learning and things like that. So
I know, right? Yeah.
Kind of soften the on-ramp is really beneficial to a lot of people.
Yeah. Yeah, that, and I hear a lot of people also with software, right? How do I even analyze this data once I get it? That can be difficult too. I didn't show it, but we have Proteome Discover has nodes with ProCite PD for intact analysis, and Biopharma Finder has a top-down node as well. But I used to do it by hand, so
yeah.
Before all that.
Just in case anyone's wondering, it ASMS coming up and May 31st, I think there's a top-down proteomics short course on one day, and there's top-down proteomics data analysis short course on the next day. So there's some really good tools there for people that are coming into the field or they want to really know what's what, because we know it's a bit daunting out there.
Those are really good courses, yeah.
Great, thank you. Any other questions, comments?
Yes, this is Paul from Bayesian Bio. And so I just wondering, and if we contact Thermo China application scientists, whether we can get that template, we can start practicing.
Okay, yeah.
If possible, and who you believe we can contact to get those templates?
Yeah, you can message me.
Okay, thank you.
Send it off to the right people. The templates are also built into your system. So if you go in the method editor, on the left side of your method editor, there's it looks like a T. There's like this funny button.
Okay.
And it looks like a T. If you click that, there's templates, but you have to be in intact mode, intact protein, to see the top-down ones. Yeah, there's a little bit of layers. That's why the new additions, they tried to make it easier to find those, because there's so many templates that there's different categories of it. So, but so there's templates there to start with as well if you look in your method editor. For the
Thanks, Amanda.
yeah, or message me.
Great, thank you. I think we'll wrap it up there. You know, people have busy days. So again, thank you to Guillaume, Rachel, and Amanda for presenting this excellent, interesting work. Thank you also to your organizations for supporting this. And we wish everyone a great day, and we'll see you October 17th with Oliver Hale talking about native top-down out of tissue. So thank you again, everyone. Goodbye.